Okay, folks, time to jump into the fun stuff. So, the citric acid cycle, the TCA, is regulated based on availability of substrate, the need for metabolites for biosynthesis, and demand for ATP. So, there are a lot of different factors kind of impinging on flux through the TCA. We'll learn about a few of those today. And we're going to start by taking a quick step before the TCA, right? The pre-TCA. Pyruvate dehydrogenase, if you check out your TCA handout, has a single-headed arrow. So you'll know that it performs a rate-determining step. And so, as a rate-determining step, it can be targeted to modulate flux. Largely, we see that the PDH complex is inhibited by product inhibition. Recall, this is a type of competitive inhibition. So, if there is a lot of product left, right, we don't need to make more. That product gets stuck in the active site and will prevent further chemistry from happening. This will drive the enzymes E2 and E3 backwards, right? It'll stop them from happening. E1 can't be driven backward. This is why it's a unidirectional reaction. We think of this as irreversible, especially for this enzyme, because remember, one of the products is CO2, and we're not just going to recapture CO2 and put it back onto pyruvate. How is the enzyme regulated? Well, primarily through the enzyme that's irreversible, E1. E1 can be regulated through covalent modification, okay? So the E1 enzyme is on or in the R state when it is not phosphorylated. The enzyme is off or in the T state when it is phosphorylated. On the right-hand side, the enzyme pyruvate dehydrogenase kinase is responsible for phosphorylating E1, and the enzyme pyruvate dehydrogenase phosphatase is responsible for dephosphorylating E1. These kinases and phosphatases, we learned already, can be regulated themselves. And so we have regulation of the kinases that then impacts how E1 itself is regulated. And let's walk ourselves through a few of these examples. So, what turns E1 on? The phosphatase turns E1 on. The phosphatase itself is activated, see the activation arrow, activated by insulin and calcium. These are both meal signals. So when you eat a meal, your levels of insulin rise to help you break down the glucose that you've just eaten. Additionally, we have a spike of calcium, again indicating that there is a need to break down the meal you just eaten and convert it into energy. So meal signals turn E1 on because we need to metabolize our meal that we just ate. Now, what turns E1 off? Well, the kinase turns E1 off, and the kinase itself is activated by NADH and acetyl-CoA. NADH and acetyl-CoA are, of course, products of the reaction that pyruvate dehydrogenase makes. If we have enough products, we don't need to make more. Also recall that NADH and acetyl-CoA are both high energy molecules. We don't fully understand yet why redox molecules are considered high energy, but acetyl-CoA contains a high-energy thioester bond. So in addition to thinking like, oh, we have enough, we don't need to make more, these are also high-energy signals. And the point of pyruvate dehydrogenase is to metabolize food to produce energy. So if we have enough energy, we don't need to make more. Now on the bottom, folks, all of these things, pyruvate, ADP, calcium, or phosphorylation of the kinase, all inhibit the kinase. And so all of these things stop the kinase from happening and leave E1 on. Why does this make sense? Well, if there's a lot of pyruvate, that means there's a lot of substrate. And if there's a lot of substrate, probably the reaction will proceed. That makes sense. Calcium, again, is a meal signal, so we should turn this off to metabolize our meal. ADP, on the other hand, is a low energy signal, right? Because we've already cleaved off that phosphoanhydride bond. So if there's a low energy signal, we need more energy. We should turn off the kinase so that E1 can do its thing. And finally, just as a note, because I know I mentioned this in a previous video, kinases themselves can be regulated by phosphorylation, and that's true of this kinase. It's turned off when it is phosphorylated. So that is how E1 is regulated, folks. We learned that E1 is regulated by a variety of things, but also note that the PDH complex itself is inhibited through product inhibition of the enzyme E2 and E3. How is the TCA regulated now, moving from PDH complex into the TCA? It is primarily regulated at its 3 rate determining steps. Again, take a look at the handout. It'll show you those 3 rate determining steps with the single-headed arrows. And the rate determining steps are regulated in a variety of ways. Of course, they're tightly linked to how much energy is required. So we'll continue to have this, are we in a low energy state, are we in a high energy state question in the back of our mind. And we'll think about the types of high energy molecules relevant to the TCA. It is also regulated, of course, by product substrate availability, product inhibition, and a new type of inhibition called competitive feedback inhibition. Let's take a look at these before we wrap up today. And let's walk ourselves through kind of each of these things here, folks. Starting at the very top, let's orient ourselves. Here's pyruvate coming into the pyruvate dehydrogenase complex. Apologies, that's not already on the slide. I'll fix that. Then we have acetyl-CoA going through the TCA. A main regulator of the TCA is right here in the middle, NADH. What do we see NADH is doing? Well, anytime you see this dotted line with a stop sign, this means it is inhibiting. So NADH is inhibiting the PDH complex, it's inhibiting citrate synthase, it's inhibiting isocitrate dehydrogenase, and it's inhibiting alpha-ketoglutarate dehydrogenase. It is inhibiting all the rate determining steps. Now, importantly, folks, you want to understand what type of inhibition might be happening with NADH. NADH is a product of pyruvate dehydrogenase, so it is probably inhibiting the pyruvate dehydrogenase complex through product inhibition. Let's take a look at the other enzymes it inhibits. First up, citrate synthase. Here is our citrate synthase, right? do we have NADH as a product of the reaction? I am not seeing NADH here, right? So in addition to performing product inhibition, we would say that it also performs feedback inhibition of some rate-determining steps. Remember, feedback inhibition is when a downstream product comes back to inhibit an enzyme. So NADH is a product of the citric acid cycle, so we'd call it feedback inhibition when it comes back to inhibit. Now, isocitrate dehydrogenase and alpha-ketoglutarate dehydrogenase both have NADH as products, so it would again be considered product inhibition. What else are we seeing here, folks? We are seeing that ATP is an inhibitor. ATP is not a product, not a direct product, I should say, of the TCA. However, GTP is. ATP goes back and inhibits isocitrate dehydrogenase. It inhibits one of the rate-determining steps, and it looks like, since it's not a product of that, it's likely feedback inhibition. Now, lastly, folks, we see succinyl-CoA performing inhibition. Succinyl-CoA is going ahead and inhibiting alpha-ketoglutarate dehydrogenase. And there's also a line, and there's also a line all the way over here, back to citrate synthase. It is inhibiting alpha-ketoglutarate dehydrogenase through product inhibition. Since it's a product of that enzyme. But why would succinyl-CoA be able to inhibit citrate synthase? It does so through a mechanism called competitive feedback inhibition. And so now I'm going to go back one slide and let's define competitive feedback inhibition. This is when a downstream product can bind the active site of an enzyme and inhibit it. So we have the feedback inhibition part because it's a downstream product coming to inhibit an enzyme, but competitive feedback because that downstream product can bind the active site. Why does this make sense for succinyl-CoA? Well, note, folks, that one of the substrates of citrate synthase is acetyl-CoA. If you go way back to the bioenergetics lecture and take a look at the line structure of CoA, you'll recognize it's this huge organic molecule. The difference between acetyl-CoA and succinyl-CoA is so small that they look more or less the same to citrate synthase. However, succinyl-CoA is not citrate synthase's substrate, so it cannot perform chemistry on it, so it is inhibited. So I'll just write CoA here. Finally, folks, it looks like we also are seeing citrate inhibiting citrate synthase, acetyl-CoA, of course, inhibiting the pyruvate dehydrogenase complex. These are each inhibiting one RDS. And these are more straightforward because we can tell that those are definitely product inhibition of the enzymes that make them. And so this is what's happening for regulation of the citric acid cycle. You see here that NADH is a main hub of regulation. It's inhibiting every single rate determining step. But we have more going on than just this. And you can also note, folks, that there are some activators on this slide as well. We see calcium activating at 3 rate determining steps. We also see ADP activating at 1 of these rate determining steps. Let's wrap it up, folks. I'll remind everyone that the TCA is amphibolic, so we won't be learning about all of this. This is what you'll learn if you take biochemistry too. But keep in mind, there's more than just what we've come together to learn. We have walked ourselves through the citric acid cycle. Pyruvate dehydrogenase complex comes before. It's a massive protein complex. We learned about some new cofactors and how they work. We learned about some similarities between the TCA and glycolysis. We focused then, folks, on the regulation at pyruvate dehydrogenase and through the TCA. With that, I'll leave you with the cat meme. See you next time.